Before diving into blood typing, we first have to understand about antigens and antibodies. And antibodies are officially called immunoglobulins. Antigens are found on erythrocytes, and antibodies are found floating around in the plasma. The antigens that you have on your red blood cells is a product of your genetics. All humans have genes for the antigens in the plasma membranes of the red blood cells. You can have genes that insert what we call the A antigen, you can have genes that insert the B antigen, or you can have a gene that says don't make any antigen. If all you have is the A antigen in your red blood cells, then you have type A blood for the ABO blood typing system. There are other systems like RH. If all you have is the B antigen, then you have blood type B. You picking up what I'm laying down? If you have the AB blood type, that means that you have the antigen A and the antigen B. And finally, if you have no antigen, if your genetics has instructions that says, no, 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 don't make an antigen, then you have blood type O. These are the types resulting from the ABO blood groups. We'll consider RH blood groups, which are positive or negative. We consider those separately. So right here, we're just going to talk about ABO blood groups. Whatever antigen you don't have the corresponding antibody. In your body, you never want the antigen and the antibody to meet each other. So if you are blood type A, you only have the B antibody floating around in your plasma. If you are blood type B, you would have the A antibodies floating around in your plasma, right? Because you would not have, again, you would not have the antibody that matches your antigen. You don't want that. If you are blood type AB, you don't have any antibodies, and that's fine. That's okay. And if you're like me, blood type O, you have both antibodies. I just want to make a note here that this is like opposite day to the previous slide, right? Like if you have blood type A, you have blood antibody B. If you have blood type B, you have antibody A. It's exactly the opposite. You never, ever, never, as the 32-point font here says, you never want your antigens on your cells to come into contact with the antibody that matches them. At least you never want that to happen in your body. We can do that in a lab with a sample of your blood. But when the antigens in your cells, when the antigens on your red blood cells come into contact with an antibody that matches them, they bond together in something called agglutination. And agglutination isn't like one blood clot. It's kind of like a million little blood clots in your blood. So in practice, in you, I never want to see blood clots. I never want to see agglutination. But in the lab, I can actually use this concept to identify blood types. So I can willfully cause agglutination in a sample of your blood in order to determine your blood type. I can intentionally expose a patient's blood to different antibodies, right? Like if you're blood type A, I could expose you to the type A antibodies, and I might see agglutination, which would tell me that you have the A antigen. If I had a serum of B antibodies, I could take a sample of your blood and expose it to the B antibodies. If there's agglutination, then I know that you have the B antigen. Right? So let's use this idea. Let's use this idea of agglutination, right? Knowing that we never want it to happen in your body, right? That's a mismatch, but that we can use this to identify your blood. I have 5 patients here, and what you see for each patient is a little tiny plate that has these wells in it, and I filled each well with the patient's blood. So first, what I can do is I can take a dropper bottle full of the A antibody, which is over here, right? And I can actually take this. Oop, that's not where I want to take this. I can take this and I can drop it into all of the A wells for everybody's blood here, right? I'm exposing their blood to a known A antibody. If I see agglutination, it means that the antibody I dropped has met with its corresponding antigen. And if I see agglutination, I can say that my patients have the A antigen. I'm not saying that all of these people have type A blood. I'm just saying that they have the A antigen. And then if I take a dropper bottle full of the B antibody and drop it into everybody's B well on their sample plates, right, knowing that I'm dropping the B immunoglobulin, if I see agglutination, then I know then the person has a B antigen. So this person has B, this person does not, Marge does not, Margaret does, Molly does, and Mary does not. These plates here, they all seem to also have a well for what's called the Rh antigen. The Rh antigen, it's simply just another protein that's inserted into the plasma membrane of red blood cells. However, the Rh antigen is inherited completely separate from the ABO antigens. They have nothing to do with each other. It's kind of like inheriting dimples and freckles. They have nothing to do with each other. So your ABO blood type is determined separately than your Rh blood type, just different genes. So if I were to take the Rh antibody and drop it into everybody's Rh well, if I see agglutination, that means that the person has the Rh antigen. And if you have the Rh antigen, then your blood type is positive. If you don't have the Rh antigen, your blood type is negative. Maria has the Rh antigen, and therefore her blood type is AB positive. Mary, over here, seems to have none. Absolutely none of the antigens. Is she dead? No, her blood type is O negative. Sorry. See if you can determine the blood type of the other three patients. And remember, agglutination tells you that the antigen is present. In the lab, I can intentionally expose antibodies to antigens, right? In like a little sample plate. However, you never want that to happen in reality inside your patient. So again, just a reminder that we use the idea of agglutination to identify blood types, knowing we never want agglutination to happen here in donation or receiving of blood. Sometimes when students do the previous slide, they get everything perfectly backwards. Like, for example, you think that Marge, whose blood type is A minus, the students think that it's B positive. If that's the case, then you have the logic of blood typing completely backwards. And sometimes looking at blood donation might help you understand a little bit why you have things backwards. So another example from the previous slide, you might have thought Maria was O negative and Mary was AB positive, but they're the opposite. So what we're about to do could help or hurt, either one. In blood donation, the general rule is that you cannot get what you do not already have. Let's apply this rule to understand who you can donate to and who you can receive from. This table only considers the ABO blood group, no positives and negatives. So if you are blood type A, you cannot receive from blood type B, right? Because they have something you don't have. If you're blood type A, you can't receive from blood type AB because, again, they have something that you don't have. So if you're blood type A, you can certainly receive from people who are blood type A, but you can also receive from blood type O people because they don't have anything that you don't have. Let's do blood type B. People who are blood type B, they cannot receive from blood type A or AB, right? People who are A or AB, they have something that the B antigen people don't have. So using our logic again, that you cannot get what you don't already have. People who have blood type B can receive from blood type B people, but they can also receive from O. Again, applying our logic, if you have blood type AB, you have everything, right? Right? You have everything. People with blood type A, they're not giving you anything you don't already have. People with blood type B, they're not giving you anything that you don't already have. And people with blood type AB, I mean, we can always donate to our same blood group. But also people with blood type O, they're not giving you anything that you don't already have. Right? Blood type AB can receive from everybody else. Unfortunately, with blood type O, you can only receive from other people who are blood type O, right? Everybody else has something that you do not have. So, in terms of receipt, it's really easy. You have to think of yourself as the blood group and say, am I getting something that I don't have? If that just made you confused, let's do this next thing and maybe something will click into place. If I am blood type A, I could not donate to somebody who is blood type B or O, right? Those blood types, they can't get my A antigen. But I can donate to blood type AB, and I can always donate to my own blood type. If I am blood type B, I can donate to people who are blood type B because I can always donate to my own. But I can also donate to people who are AB. I wouldn't be giving them anything they don't already have, right? Right. With blood donations, blood type O can donate to everyone because blood type O isn't giving anybody anything that they don't already have, right? Blood type O is not, it's following that rule that it's not giving anybody something they don't already have. And in blood donations, AB can donate only to AB, right? Because if AB is donated to blood type A, that person with blood type A would be getting the B antigen they don't have. If a person who is AB donated to someone who's blood type B, they would be giving them the B antigen that they don't already have. So with blood type AB, they can only donate to blood type AB. If we were to add in the positives and negatives, blood type O negative is the universal donor. O negative people, they don't have any antigens, AB or RH antigens. So it's like a naked red blood cell. It can go to anybody. AB positive is the universal receiver. They have everything, and they can only receive from AB positive people. So again, you cannot get what you don't already have. I guess another way to say that is you cannot give somebody something that they don't already have.